Okay. So good evening, everybody. I apologize for the postponement of the session. So that is why I am seeing the attendance is also significantly low. Ideally, I'll be taking these courses every Monday from 7 to 9. This Monday, I had some unavoidable core that I needed to complete, due to which I couldn't take the session, right? But I'll upload this one, which because of lack of attendance, it will be more of a recorded session, I guess. So I'll just go through the material that I have and I'll upload it on YouTube for you to uh, go through it, right? Without wasting time, I'll just share my screen first, right? So, yeah. So first, what we can go through is the previous year questions from week 0 and week 1 that were given in your course. And then for the latter part of the session, what I have kept is basics of molecular biology, because there were a lot of basics that were introduced during the first week of lectures. So we have not fully dived into the neuroscience part as yet. But there were a lot of concepts that we have gone through, right, in molecular biology. Because we have time over here, we will go through them in a little bit more detail. We'll still not delve in deep into that because it is not a molecular biology class, but we'll still try to cover a little more so that it refreshes your memory, right? For instance, what happened in the Michelson-Stahl experiment, what exactly happened in the Hershey-Chase experiment, and so on, right? But that will be the latter part of the session. First, what we'll be doing is we'll go through the previous year questions for week 0 and week 1. And Week 0 questions are an amalgamation from the entire course. So by the time you are through with this course, you will know the answers to most of those questions, right? That is our aim. So even if you don't know it at this point, it is okay. This is what the course is for. As and when you read more, you study more, you will understand more about the this subject, neuroscience, neurobiology, right? And then you'll be able to solve it better. So for now, let us just begin with this, right? Yeah, the first question, for instance, is the cell body of a neuron is also referred to as, the options that were given were axon, dendrite, soma, or synapse, right? So even if you are very, very new to biology itself, maybe you don't know the answer to this. But if you have studied neuron as a cell type, even in your cell biology or molecular biology courses, you will be easily able to answer this, right? We'll see what the parts of the neuron are. The answer for this one is soma. Soma itself means cell body, right? Axon is that long kind of fiber that is coming out of the cell body of the neuron. Dendrites are those tree-like or root-like structures, as you can see in this background, these things that are coming out. And synapse is basically the space between two neurons, right, where the transmission of signal occurs. I have also kept another representation of the neuron. As you can see over here, this part, this core part is the cell body, which is also referred to as soma, right? This part is the axon. Sometimes it's myelinated, sometimes it's not myelinated. We'll study about what the importance of this myelination is, or why sometimes it is not myelinated, right? And eventually we will come on to the other parts as well. Okay. So, moving on to the next question, it is action potentials are transmitted through to the cell body of the neuron from other neurons through which of these? Through axons, through dendrites, through nodes of ganglia, or through the soma or the cell body, right? So, again, we can visit the same structure and we can see that dendrites, these root-like structures that are arising out of the cell body or the soma, are the ones that actually receive signal from another neuron, right? So this is one neuron, which is a presynaptic neuron, which means this is the neuron that is passing on the signal, and postsynaptic neuron is the one that is receiving that signal, right? So the receiving end will be the part where the soma is, and which particular part will actually receive the signal? It is these dendrites, right? So that is the answer for this one. The synapse where this exchange of signal is taking place has also been zoomed in over here for your reference, and we'll study it in more detail as and when we go through the course. Yeah. So the other question is the resting membrane potential of a nerve cell is, right? So again, it is also sometimes an aptitude-based question in a lot of competitive exams, JAM, GATB, GATE, CSIR, right? So the correct answer to this is minus 70 millivolt, right? And we will, again, throughout this course, what we will understand is we know, often we do know this answer, but we don't know how this minus 70 millivolt is actually achieved or why it is this number only, right? So that we will understand over here, that it is basically an average of the different kinds of membrane potentials for different ions. It is an average value out of all of those, right? So eventually we'll study that as well. As you can see over here, minus 70 millivolt is what the resting membrane potential generally is. Then, why I did not stay in the last image for very long was because it had the answer to this question also, right? So we'll see it again in a few seconds, that the extracellular fluid has a higher concentration of dash, while the intracellular fluid has a higher concentration of what, right? So these are your options for extracellular versus intracellular. It could either be sodium and potassium, or potassium and sodium, chloride and potassium, or potassium and chloride, right? So this again has nothing much to do, or it is not very specific to neurobiology per se, right? Even in your other cell types, this is what is generally being followed, right? So extracellular fluid generally has a higher concentration of sodium, while the intracellular fluid has a higher concentration of potassium, right? That is why there is a presence of that active transport system, also Na plus K plus, which maintains this balance, right? The sodium potassium pump, as we generally call it, right? And these are the most common uh, ions that are present, right? So over here you can see that sodium in the extracellular fluid, this is the value, while intracellular is much less than that. In case of potassium, it is the almost the opposite, right? Extracellular fluid has much more than the uh, intracellular has much more than the extracellular fluid, right? Similarly, extracellular fluid also has higher amount of calcium. It also has higher amount of chloride ions, right? And many others, right? In the previous picture also, if we go, just a moment, over here you can see how it is represented is also telling you what the answer to that question is, right? Over here outside, you can see more of these pink molecules that are the Na molecules that are present, and K plus, that is the blue colored molecules over here, they are more in number inside, or the cytoplasm or the intracellular fluid, right? Uh, yeah. Then the next question is that the nodes of Ranvier are located in the cell body, or along the axon of the neuron, or the dendrites, or both the cell body and the axon, right? So the correct answer to this is that it is located along the axon only, right? Why it is only located along the axon? Because these nodes of Ranvier are basically these openings where myelin sheath is not covering the axon, right? So, in myelinated axons, there are openings at a regular frequency. These openings are known as nodes of Ranvier. And since there is no myelination itself in the cell body or the dendrites, we don't have nodes of Ranvier over there, right? Then the next one is neurotransmitters relay signals across one, the synaptic gap or the axon gap or the cell body or the node of Ranvier, right? So, which one among this is the correct answer? So, in this particular case, it is the synaptic gap, right? Synapse itself, as we were discussing in this particular figure earlier also, synapse is the region where the exchange of information is taking place, right? Between this presynaptic neuron and this postsynaptic neuron. So, this is the area where the neurotransmitters which are helping to relay this information are actually present and they are relaying signals from this neuron to this neuron, right? Thereafter, we ask another question. The central nervous system consists of which of these, right? Is it the brain and the spinal cord, or the brain and the cerebellum, or the cerebellum and brainstem, or spinal cord as well as cerebellum, right? One second. Yeah. So the correct answer to this is brain and spinal cord, right? Why? Because PNS actually consists of these two, brain and spinal cord. The other parts, other neurons that are outside or nerves that are outside of this are a part of the peripheral nervous system or the PNS. And over here, the options that are given to you apart from this is brainstem and cerebellum, right? Both cerebellum as well as brainstem are a part of the brain itself, right? It is not a separate entity. So that is why this is the most appropriate answer. So as you can see over here, this is the central nervous system, the brain and the spinal cord, while the peripheral nervous system consists of all the other nerves that are innervating the body, right? Then which brain lobe is most involved in visual processing? The frontal lobe, parietal lobe, occipital lobe, or temporal lobe. Now, this is a typical neuroscience question. So, since you have still not started understanding this neuroscience part as yet, right? I have kept this representation of the brain to first tell you what these lobes are, right? Because earlier what we used to study was the hindbrain, midbrain, forebrain, like that, cerebellum, brainstem, right? That is all we used to study. But over here, you can see that the forebrain, for instance, or the cerebrum, for instance, is also divided into multiple lobes. These four are the main lobes, right? The frontal lobe, the parietal lobe, occipital lobe, as well as the temporal lobe. Apart from that, cerebellum is there, and the spinal cord or brainstem region is present as and represented in the blue color. Right. So these are the various lobes. And you will study about this in your course. Right. Now, up in all of these, which one particular lobe is actually involved in the visual processing? Right. So the answer for that, which you will also be able to answer it very in a few weeks, is occipital lobe, right? This particular region. So this part I have kept another representation which gives the major functions of each of these lobes, right? So frontal lobe, for instance, is responsible for decision making, from for problem solving, for major emotions and behavior regulation, speech production, etc. Right? The parietal lobe is responsible for processing of senses, spatial awareness, understanding where our body is in space. Occipital, the one that is we have listed over here, is actually the major vision and visual processing area, right? Apart from this, there is temporal lobe, which is the place where memory formation, language understanding, as well as hearing occurs, right? And we have also given certain other oh, it is just a repetition of what is written over here. Cerebellum is involved in balance and coordination. You should listen to this because there is a question related to this also in this particular presentation itself. Thereafter, there is brainstem, which controls functions such as breathing, heart rate, and blood pressure, as well as sleep cycles. Right. So for now, we shift to again the cellular part of neuroscience, which are the cells that are responsible for producing myelin sheath around the neurons, right? So are these osteocytes or are these Schwann cells, oligodendrocytes, or both Schwann cells as well as oligodendrocytes, right? So the first one is osteocyte, right? The first option that is given is osteocyte. So you might already be aware, or you might know the etiology of this word osteo. It has something to do with bone, right? So anyway, you can rule this out. That osteocyte will not be the answer because it is not producing myelin. It is generating those bone structures, bone cells, right? So the other two options were Schwann cells and oligodendrocytes. This you may or may not have heard of before, but both of them are actually involved in producing this myelin sheath. Schwann cells help in the production of myelin sheath in the peripheral nervous system, while oligodendrocytes help in production of myelin sheath within the central nervous system. Right. So both these cells help in producing this myelin sheath. Thereafter, the next question is neurotransmitters travel from the presynaptic membrane to the postsynaptic membrane via which of these processes, right? Is it based on osmosis or diffusion or active transport or reverse osmosis, right? Over here, the answer is simple. There is no semi-permeable membrane or there's no membrane per se that is present within this synapse region, right? And there are no active transporters that are also present, right? So it is simply through diffusion that this process takes place. Thereafter, the other question is which condition results from the death of dopamine producing neurons in the brain? So these neurons are also known as dopaminergic neurons. Dopamine, as you know, is a primary is one of the primary neurotransmitters in the brain. It is involved in a lot of kinds of signal processing, right? It could be related to the mood elevation also, and it could also be related to the motor functions, right? Those kinds of signals also, dopamine producing neurons, like dopamine as a neurotransmitter, it performs, right? And these are certain diseases that are given over here. It is multiple sclerosis, seizure disorder, Parkinson's disease, and stroke, right? So out of these, actually, Parkinson's disease is well linked with death of these dopaminergic neurons, right? So over here, you can see a representation where this is a normal neuron, wherein the presynaptic neuron, which is the dopaminergic neuron, is able to release. So dopaminergic neuron is again a dopamine producing neuron, right? It is it has large reservoirs of dopamine within it, which it releases at the synapse, and uh, the receptors in the postsynaptic neuron are able to perceive this neurotransmitter, and it affects, or it is involved in normal movement or muscle function, right? While if there are Lewy bodies, which are like protein aggregates of some sorts, if they are present or they are affecting these dopaminergic neurons, then what happens is a condition known as Parkinson's, wherein there is very less production or release of these dopamine neurotransmitters in the synapse region, and thus the movement disorders occur, which is a key symptom of uh, Parkinson's disease, right? Please give me a moment. So I think I'll reshare the screen once. Okay. Uh, so if my cold doesn't get better, I think I will stop the class prematurely and I'll cover it in the upcoming session on Monday, right? So depending on how I long I can sustain, I'll be able to teach. Yeah. So the next question is, how do neurons transmit signals to each other, right? So do they do this via electrical signals or chemical signals or through impulses or through both electrical and chemical signals, right? So how this happens is through actually both the electrical and chemical signals. There are both kinds of synapses that are present. Sorry. There are electrical synapses also and chemical synapses also. There are more number of chemical synapses that are actually present. But as you would expect, the signal transmission will be more rapid in case of electrical synapses, right? Because the rate of flow of ions over here will be much more than the flow of ions when a synapse is present and through this means, right? But both the kinds of signal transmission actually occurs. Uh, Give me a moment. I just apply some balm. I'll share again. Yeah. So the next question is, damage to which brain region leads to loss of balance, coordination, as well as decreased muscle tone, right? So is it parietal lobe or thalamus or cerebellum or occipital lobe? So this even in our class 10th and 11th books we have studied that which is the organ that is responsible for this balance and coordination, and a while back also I told you to pay attention when I was describing those various lobes of the brain and their function, right? So what is the answer for this one? It is basically cerebellum, right? So over here you can see cerebellum is the region that is responsible for balance as well as coordination. Then Broca's area is located in which of the following? Frontal lobe, parietal lobe, temporal lobe, or occipital lobe. Now, before we understand which area of the brain it is located in, let us understand what exactly is Broca's area, right? So Broca's area is one of the areas which is responsible for speech production, basically, right? So what it means is that there was a patient in which this particular area of the brain was not functioning, or it was damaged through some way. Maybe there was some epilepsy, it was an epileptic focal point, or it was damaged during an accident. That part I don't recall very well, but it was this area of the brain that was damaged somehow. And due to that, what used to happen was the person was able to understand what the other person is trying to communicate. He could process that. He could, in his mind, he could form what has to be said. But when he used to start saying that thing, the speech wouldn't come out properly, right? The speech production wasn't happening, right? So the articulation of the production of the speech was what was affected. So that region came to be known as Broca's area. There is a contrary to this, there is another area in the brain which is located in another region which is known as Wernicke's area. Now, in that area, what used to happen was in another patient, that particular region was damaged, right? So, what used to happen is the person couldn't understand what was being said to him, the person couldn't comprehend what is being said, but the person could speak, right? But because that person can't understand what is being communicated to him or can't articulate what has to be said, he used to speak clearly, but whatever he used to speak would make no sense at all, right? Because that person has lost the skill of communication or articulation. The person can speak, but that is the skill that is lost. In Broca's area, the communication is intact, but the uh, uh, ability to produce words is what is lost, right? So and these were present in two very far apart areas. So that is how we also came to know that speech and other senses, they are basically functioning through various regions of the brain. It is not just located in one region of the brain, or one region of the brain might not be responsible for one sole purpose. Right? It might be different regions of the brain that are coming together and helping perform one simple function also, right? So Broca's area per se is located in the frontal lobe. You can see this is the Broca's area that has been highlighted over here. It is referred to as the anterior speech area, and this is the region where it is present, right? The frontal lobe area. The other area that I was talking about is this Wernicke's area, right? This is the Wernicke's area. It is present somewhere in this parietal lobe, right? Yeah. So speech articulation is one thing, speech production is one thing. All these things we came to know through these kinds of studies, right? Or reports from patients. Then what is the primary effect of myelination on axons, right? Does it protect them from damage? It slows the signal propagation. It prevents crosstalk between axons, or it greatly increases signal conduction speed, right? So which of these is actually true? It basically greatly increases the speed of signal conduction, right? So what you can imagine is this myelination is kind of like an insulation that is present, right? So if you take a wire that is not insulated properly, it will have slower speed of conduction of electrons, while if there is a wire that that is well insulated, the speed of conduction of ions electrons will increase. Why? Because over here loss is not happening outside, right? But in the naked wire, the loss will happen from while traveling, right? So that is what is the main effect of myelination on axons, right? So we hear what effect unmyelinated axon, what happens? The signal travels throughout the axon, right? In myelinated axon, what happens? It appears because it is insulated in between. It appears as the signal is jumping from one node of Ranvier to the other, right? Because within this myelinated region, there is no decay that is happening at all, or very minimal decay that is happening, right? So it appears as if signal is jumping from here to here, right? While if it is not myelinated, it will continuously decay like while propagating. Okay. Then from here comes the actual week one lectures that you have done, questions related to that, right? So, for instance, which historical period is highlighted for contribution to neuroscience? The answer to this one is 1940s and 50s, right? This was the title of one of your lectures also, who is credited with laying the foundation for chromosome theory of inheritance, right? So, which of these great scientists is, credited with laying foundation for chromosome theory of inheritance, right? Is it Gregor Mendel or Thomas Hunt Morgan or Barbara McClinton or Frederick Griffith, right? So, the correct answer for this is Thomas Hunt Morgan out of the options that are listed over here. at least, right? Along with Morgan, Walter and Sutton had also a great role to, they also contributed a lot to the chromosome theory of inheritance. But out of these, Thomas Hunt Morgan is the correct answer. So, if the white eye gene was present, right? What he saw was like this is the chromosome theory of inheritance that like from a somatic cell, the chromosomes they replicate, right? After that, they undergo a meiosis one, wherein there is reduction of chromosome number, which where they are duplicated into daughter cells, and further these chromosomal arms also separate, and then there is a further reduction of chromosomes from diploid, they become haploid over here, and that is how these various chromosomes are transmitted, right? Over here, you can see capital Y, capital R, small r, right? So these are traits. So there will be few daughter cells in which there will be capital Y, capital R that will be transferred, few daughter cells wherein small y, small r will be transferred. Similarly, few where Y, capital this is homologous chromosome. So that is the combination that we get in F1 progeny at least, right? So what experiment Thomas Hunt Morgan did? So Thomas Hunt Morgan, if you remember, used to work on fruit flies, right? So what he saw was that if the Y gene was present, right, it would always expressed in males only because they have one copy of the gene. So it was probably present in the sex chromosome, right? The males had one copy of that X chromosome while the females had two copies of those X chromosomes. So in male, if that gene for white eye was present, it would always result in white eye in the male, right? On the other hand, if the females had that gene for a white eye, then it may or may not express because females have two copies of X chromosome, and this is a recessive trait, right? So only if it is present in two copies at least of the eye of the chromosomes of the female will it express. If it is not present, then it will not express, right? So that is why it was a recessive trait, right? Uh, that basically gave insights into chromosomal theory of inheritance, and later Walton Sutton also improved that theory and presented it to the world. Then another very simple question: which system is a part of the sensory division of the nervous system? Is it vision or posture or higher motor centers or neuroendocrine system? Right. So the correct answer for this one is vision. Then what structural features of DNA did Watson and Crick describe? Did they say that DNA was double helix with complementary base pairing, or that it was single helical, or it was a triple helix with random base pairing, or it was a helix with no defined structure, right? So if you know what DNA looks like or what is its property today, you will be easily able to answer this because Watson and Crick were the ones who correctly described the structural feature of DNA, which is that it is a double helix with complementary base pairing, right? So over here you can see this one is the DNA, this is the RNA. So DNA is this is what a single helix is, right? And DNA is a double helix, right? And it has complementary base pairing, right? So G is going to, form a bond with C. A is going to form a bond with T. So that is what complementarity is. Then what is the primary role of RNA in protein synthesis, right? So does it help in replication of DNA, or it acts as a template for protein synthesis, or it transports oxygen, or it stores genetic material, right? So we can rule out the options that we think are not correct, right? Is RNA responsible for replicating DNA? No, right? Polymerase is what is responsible for that. You know, transporting oxygen is again not a function of RNA, it is a function of a particular type of protein, right? Storing genetic material, in most of the higher organisms, that is not the case. In some lower organisms, it is actually the genetic material. But over here, the most accurate answer is acting as a template for protein synthesis. That is universal in all organisms, right? So, as per central dogma, we can the actual primitive central dogma used to say you can ignore these dashed processes, and what it used to say is that DNA forms RNA, RNA forms protein, and this process is irreversible. It is unidirectional only. It can only DNA can only form RNA, RNA can only form protein, right? And this step is not reversible. It is just unidirectional, right? But in either case, RNA does act as a template for protein synthesis, right? Then what is the significance of the specific pairing in DNA, which was proposed by Watson and Crick, right? So in their paper, in that last line of their paper, what did they say? That it explains the compact structure of the DNA, or it suggests a possible copying mechanism for the genetic material, or it determines orientation of RNA, or it determines, so, okay, this option is given twice, right? But the correct answer is it suggests a possible copying mechanism for the genetic material, right? Because it was double helical, it would probably dissociate, and one of the arms of the helix, it would act as a template for, further replication, or both of the arms could also act as templates for further replication, right? So that is the possible copying mechanism that they suggested. Then the next question is which statement aligns with the central dogma hypothesis, the original one, right? Information flows from RNA to DNA and then to proteins. Information flows from DNA to RNA and to protein and cannot reverse, or the information flows only from proteins to DNA, or DNA does not participate in the information transfer at all, right? So there's only one true statement over here, which is information flows from DNA to RNA to protein and cannot reverse. This was the original central dogma, right? This is not true at this current time. There are some modifications, right? Like reverse transcription or RNA itself acting as a protein, replication happening. These kinds of additions have been, or exceptions are present, right? Then the other question is, what is the discovery which was made by Hershey and Chase in 1952? Right? So, what is the discovery that they made? Whether DNA is the genetic material, or RNA plays a role in transcription, or DNA undergoes mutation, or chromosomes are inherited independently. Right? So the actual answer is that DNA is a genetic material. What was the Hershey-Chase experiment? They basically took a bacteria and a bacteriophage. What are bacteriophages? They are viruses that infect the bacteria, right? What they did was they labeled these bacteriophages. The bacteriophage has two major components. One is the protein coat that is present, right, which they labeled with sulfur, and the one was the material within these bacteriophages, which was DNA over here, which they labeled with phosphorus, right? And what they did was they let these labeled bacteriophages infect different populations of the bacteria, right? So once the sulfur labeled bacteriophages infected the population, what they did was they first, after infection, they blended so that whatever bacteriophage coating is remaining outside of the bacteria, it dissociates, or it goes away from the bacteria. Only whatever has gone into the bacteria will remain, right? After that, centrifugation was done to pellet down the bacterial cells, and what was seen was that there were no sulfur labeled cells that were seen over here, right? So all the sulfur labeled cells went in the supernatant, or they went away, right? So they said that okay, protein at least did not go inside, right? Then they repeated this experiment with phosphorus labeled DNA, and after centrifugation, what they found was that in these pelleted cells, they had phosphorus present, right? So, what they deciphered from this experiment was that indeed, the DNA was the material that was being transferred from this viral particles to the bacteria, and it was infecting the bacteria, right? So this is the genetic material of the bacteriophage, which it needs to replicate within the machinery of the bacteria to survive and further, replicate. Then what is one structural limitation of ribose sugar that prevents its use in DNA, right? So ribose sugar is present in RNA, right? That is ribonucleic acid because it has a ribose sugar, and DNA is deoxyribonucleic acid, means it has a deoxyribose sugar, right? So the answer lies within this statement itself, right? So it cannot form bonds with bases. It its extra oxygen atom would create unfavorable van der Waals contacts. It lacks the ability to form helices, or it does not bind phosphates, right? So what happens is the ribose sugar, right? It has an extra oxygen over here, right? Which the deoxyribose sugar doesn't have. This makes the ribose sugar more reactive, right? And it forms unfavorable van der Waals contacts that makes RNA less stable than DNA, right? So that is why it prevents its use in DNA. This is the final question, which is what genetic disorder is caused by a defect in the gene for hemoglobin, right? So the options that were provided were Down syndrome, Klinefelter syndrome, sickle cell anemia, or trisomy of. Okay, sorry. I'll just stop. I'll re-share the screen, right? Yeah, so this was actually the final question also, which is what genetic disorder is caused by a defect in the gene for hemoglobin, right? So is it Down syndrome or Klinefelter or sickle cell anemia or trisomy of 21st chromosome, right? So the actual answer is sickle cell anemia, uh, which is a relatively common condition, right? Especially in some continents more than the other. Over here, what happens is that if you can see over here in normal DNA, there is this code that is present, or this amino acid, which is leucine, which is converted to this amino acid valine, right? This small change between A to U causes, or in the DNA between T to A, causes this very severe change in shape of the blood cell, which makes it, very less capable or almost incapable of carrying the oxygen or hemoglobin, right? So that is why anemic condition arises in the person who suffers from this kind of a mutation, right? It is just a point mutation, you can imagine, right? Just because of this one point mutation, the entire amino acid gets changed to valine, right? And then this kind of a condition occurs. So that is it for the first part of the lecture, right? This I have completed well in time, so we'll probably be able to go through the entire second part, which in which I am summarizing the molecular basis of inheritance. Basically, a lot of this has been picked up from the 12th, 11th, or 12th NCRT, right? It is just to brush up your concepts that we are going through this, right? For our coursework, but particularly what we'll be using is uh, this Candle, right? So Candle is a book that I am mostly following, and because Professor Varadhan, he's following another workbook, so you'll get insights from two different kinds of books, right? And there is a third one that I also follow, which is Jamie Watt's Students' Introduction to Cognitive Neuroscience. If I'm not wrong, this is the title of the book, which is also a very simple book to study, right? So those are the books we'll be uh, looking at once we are uh, actually studying neuroscience, right? Over here, this is just a molecular basis of inheritance to brush up your concepts before we dive into, hardcore neuroscience. Major things that we'll be studying are structure of DNA, how it replicates, the process of making RNA from DNA, which is known as transcription, the genetic code, what is the genetic code, how the sequence of amino acids basically determine this genetic code, or determine what kind of protein is being formed, the process of synthesis of these proteins, which is known as translation, and the elementary basis of the gene regulation, right? So first off, we understand what is nucleic acid, right? Yeah, please give me a moment. Yes. So yeah, I'll share again. Okay. Yeah, I've applied the balm and it is really irritating me right now. So what exactly are nucleic acids, right? So nucleic acids are chemical compounds that store and transmit genetic information in cells, right? They are composed of these major components, which are bases, sugars, as well as phosphates, right? So you can see that these are the various parts of which they are composed of. The sugar, which could be over here, it is deoxyribose. As you can see over here, there's a phosphate group, and there are nitrogenous bases, which could be adenine, guanine, cytosine, thymine, thymine, or uracil, right? And these are the various kinds, right? So this is again these are two kinds of ribose sugars, for instance, that are there. It could either be deoxyribose, which has only a hydrogen group over here, or a hydroxyl group containing ribose sugar, which is actually present in RNA, right? There are these bases, which I told you could be either of these, and there are phosphate groups, right? And this is how they are attached. Okay. So, the deoxyribose nucleic acid, that is DNA, and RNA are two types of nucleic acids that are actually found in the living system, right? These are the representations of this. This is single helical and this is double helical, right? Uh, RNA has adenine, uracil instead of thymine, guanine and cytosine, and over here it has thymine instead of uracil, right? That is a major difference apart from the ribose and deoxyribose sugars, right? So DNA is a long polymer of deoxyribonucleotides, and the length of DNA is usually defined as number of nucleotides or nucleotide pair that is present in it. It uh, this is just about the DNA, lots of characterization of DNA. Hold a moment. Yeah. Yes. So there are various characterization of DNA which we'll not get into. Right. We have already gone through this. What is the structure of a polynucleotide chain? It contains these three kinds of things, right? There are two types of nitrogenous bases that are present. They could either be purines that are adenine and guanine, which are those double, they have a hexose and a ribose, and the pyrimidines, which have just that hexose part, right? Then cytosine is common for both DNA and RNA, but thymine is present in DNA, which is basically complementary to cytosine, and oh sorry, it is common to adenine actually. And the uracil is present in RNA, which in the place of this thymine, right? Actually, adenine and guanine are also common in both DNA and RNA. So this is the actual structure of all of these nucleic acids, which for the sake of this course, at least we do not need to understand, or we don't need to remember also, right? So we'll skip the parts that are not very necessary for us. Uh, just to brush up your memory, the two nucleotides, this is one nucleotide, this is another, they are linked through a 3,5, right? 3,5 phosphodiester linkage to form a dinucleotide, right? More nucleotides then can be joined in this manner to form a polynucleotide chain, right? A polymer thus formed has one end free at the 5' moiety. This is the phosphate end that is free, and one dash at the the 3' end, which is the hydroxyl moiety, right? And the backbone of the polynucleotide is formed due to sugar and phosphates. You can see that inside, towards the inner side, is the are the these nucleotides, the adenine, thymine, guanosine, or cytosine, the backbone is actually being formed by either the sugar and the phosphate groups that are present in between, right? So these nitrogenous bases that are present in the middle, they are linked to these sugar moieties, which are forming the backbone of the DNA and RNA, right? Now, what are the major differences between DNA and RNA? One is the presence of thymine in DNA and uracil in RNA, right? Apart from that, this 2-OH is present in case of RNA and it is absent in case of DNA, right? That is the other main difference. Then this was how the initial stages of understanding of this nucleic acid or DNA was actually initiated, wherein Frederick Wester in 1869 itself said that DNA has an acidic substance present in the nucleus, is an acidic substance that was present in nucleus, and it was he identified it and named it as nuclein, not DNA but nuclein, right? Just a Thereafter, this double helical structure of DNA was later identified, right? It was credited to James Watson and Francis Crick, but as you have already gone through in your lectures, they had not done any actual experimentation, right? Their model of how DNA structure is or what it is was based on the experiments of the X-ray crystallographic experiments that was done by Rosalind Franklin under the guidance of Morris Wilkins, right? So they were the majorly credited towards finding that DNA is actually a double helix structure, but it would not have been possible without the experiments that were done by Morris Wilkins and Rosalind Franklin, right? Then one of the hallmarks of their proposition was base pairing between two strands of polynucleotide chains, right? This base pairing is complementary as we know it now, right? So we'll not get into detail of this. And how did we understand that this was actually complementary, right? That was actually proposed based on the observation of Erwin Chargaff, right? That is why this complementarity is like you can count how many adenines, guanosines, cytosine or thymine is there based on even knowing one of them, right? So that is because the presented a principle which is now known as Chargaff's rule, which says that the ratio between adenine and thymine and guanine and cytosine are constant and it equals to one, right? So this was the Chargaff's rule. Then there is complementarity that is present in DNA strands. The base pairing confers very unique property to polynucleotide chains. They are said to be complementary to one another, and therefore, if the sequence of bases in one strand is known, then automatically we can determine what is the sequence that is going to be there in the other strand, right? Yes. Uh, Yes. So the base pairing. So if we know the sequence of one strand, for example, this is the main strand, we will be able to understand which is the complementary strand, what is the base pairing over there, right? So in front of A, there will be T, in front of T, there will be A, then G will have C in front of it, A again, T will have A and A, G will again have C, right? So that is how it will happen. So we'll see that actually is the case, right? So there is a TAC, TAAC, like we were saying, and so on, right? So this is how the complementarity works over here. So if each strand from DNA, which is the parental DNA, acts as a template for synthesis of a new strand, then two double stranded DNA daughter cells will be formed, daughter DNA will be formed, and it will be whatever is produced will be identical to the parental DNA molecule, right? So that is how replication happens in DNA. Okay. Then what are other features of this DNA double helix? That it is made of two polynucleotide chains. These chains are anti-parallel to one another, right? That is not visible over here. We will see that this. So they are anti-parallel in polarity, meaning that one chain has 5' to 3' polarity, the other has 3' to 5'. You can see that over here, this sugar is facing upward, over here, sugar is facing downward because they are anti-parallel, right? So, bases in both of these two strands are paired through hydrogen bonds. These hydrogen bond pairing is two hydrogen bonds between A and T and three between G and C, right? That is what is written over here. always the case will be that one purine is paired with a pyrimidine, right? So that is the case with A also and G also, which are purines. They are always paired with pyrimides, which are T and C or U and C, right? Then, okay, I had already postponed this class once, so I didn't want to do it again. But it is really hard to take the class at this kind of a condition, right? Okay, again, these are characterizations of DNA, which we don't need to get into right now for the understanding, right? Uh, okay, so this also will not get, we'll begin with central dogma now, right? So who proposed central dogma? It was Francis Crick. In his book, he proposed the central dogma, right? What he said was that genetic information flows from DNA to RNA to protein, right? So this is what happens, is what he said. And he also mentioned that this is the only direction in which it will flow. It will not at all be, it will be unidirectional. It will not go in the reverse direction at all, right? Okay, there is some repetition over here. Okay, I think there is one file that is missing in between. Just give me a moment. Yeah, yeah, okay. There were some files that were missing. I have found them. I'll just present the screen again. Yeah, okay. Okay, again, similar kind of a problem I'm facing. Yeah, yeah. Now it's fine. So now we know that that is not the case. Central dogma has been proven incorrect, right? Or there have been certain modifications that have been made to it, rather. For instance, there is a kind of RNA that acts as a protein itself. It is known as a ribonucleoprotein, right? The RNA can also form DNA through reverse transcription, right? So it defies the unidirectionality of the original central dogma that was proposed. So these are some exceptions that have been made, right? Then the question arises that Yes. Yeah. So the next question that arose was that the DNA is such a vast material, right? So how is it present within just the nucleus of the cell, not even the entire cell it is covering, right? So that is where this concept of packaging of the DNA helix came, right? We'll not go into very much depth of this also, but we'll just go through the concept once, right? So, DNA is somehow packaged to fit into that size of the nucleus itself, right? So, how is such a long polymer packaged within a cell, right? So, in prokaryotes, they do not even have a defined nucleus, right? It is scattered throughout the cell, right? This is what it looks like. So over there, it is known as a nucleoid and not a nucleus per se, right? So it is held with some proteins that has a positive charge because DNA, as we know, has a negative charge, right? So this nucleoid is organized in large loops by many proteins, right? In eukaryotes, we know that these positively charged proteins are basic proteins known as histones. Right, so these histones are rich in basic amino acid residues like lysine and arginine, and they really help package the DNA because they allow this negatively charged DNA to wrap around its positively charged core. Right, so that wrapping helps in packaging of the DNA. Right, so both the amino acids, lysine and arginine, are positively charged in their side chains. So they organize to form a unit of 8 molecules, which is known as histone octamer. Right, this is what it looks like. There are two H3 and H4 and two H2A and H2B. Right, that are present, which come together for first as a tetramer. This H3, H4 form a tetramer, then H2A, H2B form a tetramer, and they come together to form an octamer, right? And the DNA, as you can see, is wrapped around this octamer of histones. So this is how packaging of DNA works, right? So this is a double helical DNA. It is packaged on these histones, which in turn are packaged in very complex forms. It forms a fiber-like structure, and the fibers further are very compactly and condensed form it is present in, which are known as chromosomes, right? So these histones are the DNA wrapped around histones looks like this beads on string structure under electron microscope, right? Then, uh, okay, we can just skip this for now. It is going into too much detail of packaging, right? Then we understand those experiments that you have heard through in your first week of lecture, which is the search for genetic material. What were the questions that came, how those questions were answered, what were the experiments that were performed, and who actually performed them, right? So by 1926, that is around still primitive to when we started full-fledgedly understanding about the brain, right? So first, these things were happening, right? So by 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level, right? So before this, people had said that there were traits that were passed on. Maybe there is some material that is helping in passing on this trait. Then uh, Morgan, Walter, Sutton, they had come to the level of these chromosomes also. But now at the molecular level, we wanted to understand how it was happening, right? So previous discoveries by Gregor Mendel, Walter Sutton, and Thomas Hunt Morgan uh, and numerous other scientists had narrowed the search to chromosomes located in the nucleus of most cells. Right now, what if this chromosome is actually the transforming principle, or what is the genetic material? Right. So, first set of experiments came. In 1928 by Frederick Griffith, right? So he worked on Streptococcus pneumonia, which is a kind of a bacteria, which is responsible for causing pneumonia, right? And he witnessed a miraculous transformation in the bacteria, right? What he did was, during the course of experiment, there was a living organism, bacteria, which had changed in physical form. How he found this out was that when he introduced this bacteria on a culture plate, there were some colonies that were smooth and shiny, which were labeled as S bacteria, and others which were rough or R bacteria, right? What he found was that this S bacteria had a mucus coat, which is a polysaccharide coat, while the R strain does not have this coat, right? So it made this S strain more infectious, right? So this was the virulent strain. So if the mice were infected with this S strain, they would develop a pneumonia infection and they would die. But the mice that were infected with R strain did not develop pneumonia because it was a non-virulent strain, a strain of that bacteria, right? And they would live. What happened was that this is a visual representation of what we said. The smooth strain, which is virulent, if that was injected in the mice, it dies. But in case of non-virulent R strain, the mice lived, right? What he did was, one second. Okay, so what he did was that he used this heat-killed smooth strain. That was the virulent strain. So when he heated it, the virus died, right? And then when he injected it into the mouse, the bacteria had died. So the bacteria were not alive. So the mice still lived, right? But what happened was that when he introduced a rough strain, which had also contained the heat-killed smooth strain, right? So he mixed two strains, which had the heat-killed smooth strain and the rough strain, right? So in this case, also the mice was not dying. In the rough strain case, also mice was not dying. But when he injected them together, the guy still died, right? Although the virulent strain was inactive over here, right? So what he thought was that So what he thought was that somehow the R strain had been transformed by heat-killed S strain, right? So there would probably be some transforming principle which transformed from the heat-killed S strain and enabled R strain to synthesize a polysaccharide coat and become virulent, right? Because he recovered living S strain bacteria from the dead mice, right? But there were two living S strain bacteria that he injected, right? So that is why he thought that there was some principle that transformed this R strain to S strain, right? And that has come from the heat killed S strain itself, right? So he said that this must be due to transfer of genetic material from the S strain to R strain. However, he did not know what this genetic material was, right? But now we know that there is something that can transform one strain into the other strain, and this is because of presence of some kind of a genetic material, right? Then at that time, we knew that there were three candidates that were the candidates for genetic material. They were proteins, DNA, and RNA, right? So everybody they were very interested in finding out which of these were was actually the transforming principle, right? So what happened was most of them were in favor of a protein being the actual genetic material, right? Because protein is the end product. Every kind of enzyme, every kind of end product that is doing stuff in any case, any biological system, that is actually a protein, right? So most people were convinced that it is actually a protein that is responsible for being the transforming principle. One second. Yeah, I'll share the screen. Yeah. So, then came the study by Avery, McCarthy, and MacLeod, which actually said negated protein as a candidate for being the transforming principle, right? So they worked to determine biochemical nature of this transforming principle in Griffith's experiment. What they did was they purified biochemicals, which were proteins, DNA, or RNA from this heat killed S cells to see which one could transform live R cells into cells, right? Into S cells. So what they said was that they, from this heat killed S cells, they first purified only protein, then only DNA, then only RNA, right? And separately, they gave it to separate colonies of R cells, right? In one colony, they gave only protein. One colony, they gave only DNA. One colony, they gave only RNA, right? To see which colony develops into the S cell colony, right? So they discovered that DNA alone from S bacteria caused R bacteria to become transformed into the S type bacteria, right? So this is again a representation of that, right? So when they were adding uh, uh, only DNA, then only it could uh, form this S cell, right? When there was no DNA, that S cell would not appear. So they concluded that DNA is hereditary material, but still not every biologist was actually convinced, right? So then actually came the Hershey-Chase experiment, right? The one that we have spoken about in the previous year questions also, right? So what Hershey and Chase actually did was they worked with viruses that infected these bacteria, which were known as bacteriophages. Now they also, through another approach, proved that DNA indeed is the genetic material. What they did was that bacteriophage generally attaches to bacteria and its genetic material enters bacterial cell. It treats the viral genetic material as if it was its own and subsequently manufactures more virus particles, right? So virus genetic material basically takes help from the bacterial replication machinery to replicate, right? So over here, this is generally what happens in case of a bacteriophage, right? So this is a bacteriophage, and it will inject its genetic material, which will go. It will take help from the replication machinery of the host, that is a bacteria. It will form more viruses, and then it will basically rupture this bacteria which it used to replicate, and more viruses will be released, right? So that is how it functions. What they did was that they basically grew these viruses on a medium that contained radioactive phosphorus or radioactive sulfur. So if it is radioactive phosphorus, we know that there is phosphate backbone in the DNA, right? So it will go and it will bind to DNA. But if it is radioactive sulfur, it will go and it will bind to the protein coat, which has some amount of sulfur in it, right? So the viruses grown in presence of radioactive phosphorus contained radioactive DNA, but not radioactive protein because DNA contains phosphorus, but protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein, but not radioactive DNA because DNA does not contain sulfur, right? So these radioactive phages were allowed to attach to E. coli bacteria. Then what was done was they were allowed to go through the infection cycle. After that, this mixture was agitated through which whatever was outside of the bacteria would dissociate, right? And then they were put in a centrifuge, and this was the result that was obtained that we have already gone through. That no radioactive sulfur was detected in bacterial cells, but radioactive phosphorus was detected. So this means that DNA is the genetic material of these bacteriophages also that has gone into the uh, bacteria and not the protein, which has dissociated itself, right? So bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was a material that was passed from virus to bacteria, right? So bacteria were infected with viruses that had radioactive proteins, were not radioactive at all, and this indicated that proteins did not enter bacteria from viruses. Therefore, genetic material that pass from virus to bacteria is DNA, right? So after this experiment, which was an add-on to the Avrion McCarthy-McLeod experiment, it was established as a fact that DNA acts as the genetic material, right? However, later we found out that several viruses also has RNA as a genetic material, right? But then the questions arose that why DNA is a predominant genetic material and RNA is not, right? And when we have studied about the differences between DNA and RNA, that basically helped solve this doubt, right? So we know that uracil is also more reactive and less stable than thiamine. And this hydroxyl group is also more reactive and less stable than the hydroxyl group that is present in the DNA, deoxyribose sugar, right? So because of these two very reactive groups present in RNA, it is less stable, which is why DNA is the principal genetic material and not RNA, right? Because a good genetic material would not mutate very often, right? So it should fulfill these criteria that it should be able to generate its replica, which DNA can do. It should be stable chemically and structurally, which is more, describing DNA than RNA. It should provide the scope for slow changes or mutations that are required for evolution, right? It is not that DNA, there is no mutation at all that can occur, right? It can occur, but it is relatively slow, right? It is not fast changing like RNA. So RNA viruses, for instance, coronavirus, it changes so often, right? It should be able to express itself in the form of Mendelian characteristics also. If one examines each requirement of the rule of base pairing and complementarity, both DNA and RNA have ability to direct their duplication, uh, and proteins fail to fulfill the first criteria itself, that is replication it cannot do, right? But genetic material should be stable enough, right? And DNA is more stable than RNA, we know that. uh, so yeah, that is how it has been proved that DNA is a better genetic material, right? But which one do you think was the first genetic material? Was it DNA or RNA? Right? This is just a curiosity question, right? So it was probably RNA, right? Because the primitive organisms generally have RNA as a genetic material, right? That is why this theory is also known as RNA world, because RNA was the first genetic material. There is enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc., they evolved around RNA. Right? That is why still DNA can directly not form protein. It still requires an intermediate stage RNA to form proteins, right? So RNA used to act as genetic material as well as a catalyst. But because it was a catalyst, it was more reactive and unstable. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable, right? And it being double-stranded has complementary strands. Further, it resists changes by evolving a process of repair, right? So, the initial models of DNA replication that were given were either it is semi-conservative, which means one second. Okay, I'll share again. So actually the major portions or experiments that I wanted you to recollect, I have covered. So let's see how long I can actually sustain the entire thing. I am just thinking we'll finish it, but we'll see. right? So once we knew that is indeed the genetic material, we wanted to understand how does DNA replicate itself, right? So there were three models that were proposed initially. One was a semi-conservative model, which said that there is a parental DNA strand. Because it is a double helical structure, the two helix separate, and each of the helix acts as a template for a new helix. And this is how the replication keeps happening. The second thing that was suggested was a conservative model, which said that there is a parental strand of DNA. The two strands separate and they act as template for the new strands, but they form one kind of like they form one they again combine and form one strand, and the two new strands that had been formed through replication form another DNA molecule. Just a moment, please. Yes. Yeah. The third model that was suggested was the dispersive model, right? What does this dispersive model says was that the parental strands, they randomly break into multiple pieces. These pieces, they in front of them, whatever piece is present, that develops through complementarity, right? And the new DNA that is formed, it contains parts of the parental strand and parts of the new strand. That is what it said, right? So in this original parental strand was retained and new strand was formed separately. In this, there was partly a parental strand and partly a new strand was formed, right? So these were the three models that were proposed: the semi-conservative model, conservative model, and dispersive model, right? Out of this, how was it checked which model is actually correct, right? So, what Watson and Crick had proposed as their original statement was that it has not escaped our notice that specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material, right? So, this is what was the last line of their paper. So, the scheme suggested that two strands would separate and act as template for synthesis of new complementary strands. That is, semi-conservative would probably be the most reliable kind of thing, right? So, after completion of replication, each DNA molecule would have one parental strand and one newly synthesized strand. So, this semi-conservative made most sense. But how was it proven, right? So, to prove this, it was shown in E. coli, another set of bacteria, and subsequently in higher organisms also. So, it was done by Michelson and Stahl. It is known as the Michelson-Stahl experiment. It was performed in 1958. What they showed was that they grew E. coli in a medium containing radioactive or heavy isotope of nitrogen, N15, right? Which is the only nitrogen source for many generations. So many generations kept growing in this only. Later, what they did was that this heavy DNA molecule could be distinguished from normal DNA by centrifugation in cesium chloride density gradient, which means that once this heavy density, heavy isotope of nitrogen is centrifuged, it will lie below the N14 or the other isotope or lighter isotope of nitrogen, right? Second, what they did was after growing the E. coli for many generations in that N15 heavy isotope, they transferred it into a medium with normal nitrogen or N14, which was a lighter isotope, right? Then they separated Then a few cycles, they let these cells divide over here also. They took samples, various definite time intervals as the cells multiplied and extracted DNA that remained as double-stranded helices, right? Then what they did was the DNA that was extracted from culture, one generation after transfer from N15 heavy isotope to N14 normal isotope, what they saw was that the hybrid of intermediate density was obtained, right? So I will show it to you. It will be easier to understand. So earlier only this heavy isotope was found, then, after changing it into that N14 medium, what they saw was after 20 minutes, that is first replication cycle, when they collected the media, what they saw was that there was an intermediate DNA of N14, N15 that was present, right? And after two cycles, if they collected, after around 40 minutes, when two complete cycles of replication of E. coli would have occurred, what they saw was that there were three different kinds that they saw. There was an intermediate kind which had N14 and N15. There was a lighter kind that had, so two different kinds actually. It's because only N15 is no longer available, right? So there was an intermediate kind hybrid that was present and a new lighter kind N14 that was present because now it is in an N14 media, right? So they determined that yes, semi-conservative is actually the kind of replication that is happening, right? So yeah, that is how it was formed. Then there is this replication machinery. So we'll stop at this. The major portion that was spoken about at least in the lectures, the Hershey-Chase experiment, Michelson-Stahl experiment, before that Avery McLeod-McCarthy experiment, and part of how DNA was discovered, right? All that has the Griffith experiment, original one, right? All this we have discovered that discussed already. I hope it has refreshed your memory up to a certain extent, right? So these are concepts of basic molecular biology and somewhat cell biology that uh, form the basis of the fields that have come after that, right? You will not actively need them in your neuroscience course, but it is always good to brush up your concepts because neuroscience is not a completely independent field that has come up on its own, right? uh, Everything that we have studied post this 1940 1950 was after this DNA was discovered, right? Somehow, at the molecular level, even neurons have some kind of DNA, right? The function is what is different, and that is what we will study in detail at length in the rest of our course, right? So I sign off. Just to remind you, all my subsequent lectures will be on every Monday between 7 to 9 p.m. Right? I hope to see you there, and uh, I hope that there will be a lot more attendance. Otherwise, you will have these uploaded lectures at least. So all the best for your course. I hope you learn well, right? So I'll stop the recording and then we can leave the session.